All right, time to go teach. Hello and welcome to this next episode of Tutors, where we are continuing the endomembrane system in the cell, and we are looking at what happens within the nucleus. Specifically, we are going to be looking at the process called transcription. Let's get this thing started. Targets of the day. The first target is to identify the main molecules that are involved in transcription. Next is to then describe what the functions of each of those individual molecules will be in the process of transcription. And lastly, we are going to look at explaining why the resulting mRNA strand is not exactly complementary to the template DNA strand. There are going to be some edits that are made to ensure that it's going to be a little bit different. So let's move to how and where this all happens and what is being used. First off, remember that this is all happening in the nucleus. So here's our nucleus right here. And on the inside of our nucleus, we have the DNA. And the process of transcription is going to take place in 3 different stages. Those stages are initiation, elongation, and then termination. It uses the DNA that happens to be inside the nucleus as your template, and the DNA for eukaryotic cells is separated into different strands. Each strand of DNA is called a chromosome. But while the DNA is inside the nucleus during this part of a cell's life, that DNA is unraveled or it's decondensed, and it stands in this form called chromatin, where we cannot differentiate between the different individual strands. So all the strands are basically unraveled in some ways. It's sort of like a bowl of spaghetti of DNA. Yeah, there's individual pieces of spaghetti, but it's very hard to tell which strands are which when they're all mixed up together. The molecules involved, well, first off, obviously, we just talked about it, is DNA. So DNA is the first molecule that we can look at, and the DNA is going to serve as a template for the RNA strand that is going to be completed. But DNA is made up of 2 strands. So the first strand that we can look at is the template strand, but the other strand, the opposing strand or the complementary strand, that is referred to as the coding strand. Next, we happen to have the enzyme that is going to build our RNA polymer, and it is RNA polymerase. And then to make up the actual RNA molecule, we have the RNA nucleotides that will be put together to form our macromolecule. The section of DNA that is going to be used as a template for our mRNA strand is called a gene, and that contains our unique sequence that we need to code for our specific polypeptide, which is the end result of transcription and then translation. Right now in this lesson, we're only looking at transcription. Translation is in the next episode. And each gene, they have their own initiation site. They have a unique initiation site for that specific gene. And then proteins are going to come in, and they are going to separate out our 2 strands of DNA, the template strand and the coding strand, so that those nucleobases on the template strand are now free and accessible to RNA polymerase. RNA polymerase 2 is going to move along the template strand in the direction of the 3' end toward the 5' end. As it's moving along, it is going to take the nucleotides, the RNA nucleotides that are complementary to the template strand, and add them to each other, forming our full, complete RNA strand. This process is called elongation because our RNA strand is getting longer. Let's look at this process in action. So first, we know that our DNA is going to have to open up to allow for the RNA polymerase to enter in. Once the RNA polymerase has entered in, it is going to move along the template strand in the 3' toward the 5' direction. Remember, that's based upon the orientation of that deoxyribose sugar going from the orientation of the third carbon facing one direction and the fifth carbon facing in the exact opposite direction. And that means that our RNA polymerase is going to move from the right side of our screen to the left side of our screen. It's going to bring in the monomers for our RNA and bond them together. And it will continue moving right along the DNA template. The mRNA is going to break away from our DNA template, and our DNA is just going to hydrogen bond right back together. And our RNA polymerase just moves right across, unwinding and unzipping the DNA so it can continuously build our strand of RNA. Once RNA polymerase has completed transcription of the specific gene, it will come across a specific sequence of nucleotides on the DNA, and that will force it to terminate the process of transcription. The newly constructed RNA strand, though, is not complete. This strand is complementary to our template strand. It is also recognized anti-parallel to our template strand, just like the coding strand is. The difference between our RNA strand and the coding strand is that instead of having the nucleotide thymine, we now would have the nucleotide uracil. And instead of having deoxyribose sugars, it only has ribose sugars in its backbone. But that's not the only difference between the RNA and the coding strand. In fact, now we are going to have to go into a step called RNA processing. And in RNA processing, what's going to happen is there are going to be additions added to each end of the RNA molecule. The purpose of these different additions, the 5' cap and the poly A tail, are to protect the RNA that is in the center, the actual sequence that the cell wants to have translated out in the cytoplasm. But there's too many nucleotides in the center right now. In fact, there's extra material there. There's extra RNA, and that RNA has to be removed. The RNA that needs to be removed is called the introns. And so the introns, which are shown here in yellow, they will be removed. They will be cut out, and the RNA exons will be spliced back together. They will be joined together. And now we have our fully formed RNA molecule. This specifically would be mRNA or messenger RNA. The messenger RNA now is going to exit the nucleus. Not all RNA necessarily will exit the nucleus, but mRNA will exit the nucleus, and it will have to leave through a nuclear pore. So that way it is now in the cytoplasm where translation can begin. So now why don't you give this a shot? If this was your strand of DNA, and you knew that this top strand was your coding strand, and this bottom strand is your template strand, what would the mRNA look like that would be constructed? All right, so now you should have your mRNA transcript all written out, and it should look something like this, where now it is a complement of the template strand. And don't forget the 5' end and the 3' end being anti-parallel to the template strand. Again, looking very similar to the coding strand, the only difference in this case being that we have uracil instead of thymine as a nucleotide. And summary of all this. Recognize all our cells, they contain DNA. And the purpose of that DNA is it's going to provide those instructions so that the cell can synthesize polypeptides. It's also the cell can make that working biomacromolecule, the protein. And through the process of transcription, that's how the cell is going to make RNA, which is the messenger that is going to be used by ribosomes to build your polypeptide. The pre-mRNA is the mRNA that is made that hasn't yet had those introns removed. Once the introns are removed, you have the final product of mRNA that is going to be used by the cell. And mRNA, that is going to carry the instructions out of the nucleus and into the cytoplasm so you can synthesize your polypeptides by the ribosome, which we will talk about in our next episode on translation. Until then, be awesome, stay awesome.